Good morning. Welcome to the last New South Wales Science and Research Breakfast seminar of what has been an interesting and difficult year, I think, for everyone. I'm very, very pleased to welcome Professor Antoine van Oijen from the University of Wollongong to talk about antimicrobial resistance. I want to begin by acknowledging the traditional owners on the land on which we are meeting today jointly. For me, it is the Gadigal people of the Eora Nation, and I wish to pay my respects to their elders past, present, and emerging. With Parliament House still only permitting live events in small numbers, we've made the decision to wait until 2021 to attempt to hold these events in person. The 2021 seminar calendar is being developed and will be shared shortly. Although I will note that certainly in Sydney, it's interesting to observe how things appear to be getting back to normal with people in the streets, people in restaurants, and people, in fact, taking part on Circular Quay in what looks like to be a nice summer day. Can I note that to submit questions today, please use the chat function on your screens on the right-hand side. I will ask these questions to Antoine at the conclusion of the seminar. I last met Antoine just a few weeks ago. He was the recipient of the 2020 Premier's Prize for Science and Engineering for excellence in medical and biological sciences. Distinguished Professor Antoine van Oijen is director of the $80 million Life Sciences Research Institute, Molecular Horizons, at the University of Wollongong. Antoine's research investigates antimicrobial resistance, which occurs when bacteria and other microorganisms become resistant to antimicrobial drugs, including antibiotics. The alarming reduction in effectiveness of antibiotics caused by evolutionary changes in bacterial populations under pressure from antibiotic overuse makes the treatment of infections difficult, costly, and sometimes even impossible. The future impact of superbugs worldwide on human health is still largely unknown, but is projected to eclipse the health burden caused by cancer and cardiovascular diseases. Armed with the centre's two powerful electron microscopes, Antoine and his group develop molecular imaging approaches to understanding how bacteria evolve resistance and use this knowledge to develop new drugs and new diagnostics. Interestingly, he pairs this molecular approach with population health programs that he has developed to track how superbugs travel through the community and our hospital system. Antoine has had a distinguished academic career, leading research groups at Harvard Medical School and at Groningen University in the Netherlands, before moving to the University of Wollongong in 2015 as an Australian Research Council Laureate Fellow. In addition to his ARC Fellowship, Antoine has received prestigious fellowships from the US National Science Foundation, the European Research Council, and the National Health and Medical Research Council. As I mentioned, this year, Antoine also won the New South Wales Premier's Prize for Excellence in Medical and Biological Sciences. Please join me in welcoming distinguished Professor Antoine van Oijen from the University of Wollongong as he presents Tackling Antimicrobial Resistance from Molecule to Community. Over to you, Antoine.
Thank you, Hugh. Thank you for this very kind introduction. I'm obviously also a bit sad that I'm not able to see my undoubtedly wonderful audience face. Can people hear me now? Great. That was weird. Anyway, I'm not sure whether I was inaudible from the beginning, but again, I would like to thank you and hope to be able to provide an engaging hour for you from the comfort of my home, and sadly not face to face. My entire career, I've been interested in how complex molecular systems work, and in particular, complex molecular systems that are important to support life. And in the last few years, I've grown more and more interested in antimicrobial resistance. As you explained, how do bugs, microbes become insensitive to the drugs that we in the health sector used to combat infections. And I'll use this hour to take you on a journey showing you how we develop physical tools to visualize how at the molecular level processes work that are related to this antimicrobial resistance evolving. And I'll show you a bit of experimental data that, I have to warn you, is a bit scary. Shows you how quickly bacteria can become insensitive to antibiotics. And I want to tell you a bit about the population health work that we started doing with a wonderful team of collaborators, trying to better understand how antimicrobial resistance travels through the community. So essentially what I want to do, I want to show you how we can really tie the molecular and the cellular in with the community level approach of understanding important health problems. What I would call a real holistic approach to solving 21st century health problems. And in my mind, antimicrobial resistance, or AMR for short, is probably one of the scariest 21st century health problems for which as a society, I think we're really not prepared for to tackle. I want to start my story by telling you about this young man, Alexander Fleming. He is the one who discovered the first antibiotic, penicillin. And the story is quite interesting. He was a microbiologist and he worked in his lab. In 1928, he at some point went on a family vacation for a few weeks. And he was known actually to be quite a sloppy lab scientist, not the tidiest. What happened is that he left a few plates with microorganisms in the windowsill and came back 2 weeks later and realized that he had all of a sudden gotten contamination in his plates. And he had both bacterial and mold contaminations. And the one thing he saw is that the mold on his bacterial plates prevented the bacteria to grow. So you already started to see there that the mold must have been producing a compound that kills the bacteria. And then one interesting anecdote is that supposedly he looked at his plates upon returning from vacation and he said, that's funny. And this is one of the lessons I think is really important in understanding science. When researchers discover something profound, they actually don't run around and yell, Eureka. They actually typically say, that's funny. When you hear a scientist saying that's funny, then you know they're on to something potentially quite interesting. So in 1928, he discovered penicillin. In the early 40s, 1940, a couple of scientists, including the Australian Howard Florey, worked together to massively scale up the production of penicillin, because very quickly it was recognized that it was quite a powerful agent to combat bacterial infections. And in 43, I believe, it was already rolled out at scale onto the European battlefields and played an enormous role in saving hundreds of thousands of lives of soldiers, Allied soldiers in Europe, from infections because of combat wounds. And I think it's fair to say that antibiotics are really one of medicine's greatest triumphs. The estimates are that roughly 100 million lives have been saved since the moment penicillin was discovered and started to be used, using all kinds of antibiotics to cure and treat simple infections that before 80 years ago probably would have been lethal. However, sadly, this is not the case anymore. Because of evolutionary processes that I'll tell you more about in the rest of my talk, bacteria and other microorganisms like yeast have become more and more resistant to antimicrobial drugs or antibiotics in the case of bacteria. And there was a hallmark report that was published a few years ago, commissioned by the UK government, where a panel of scientists chaired by Jim O'Neill, who used to be the chairman of Goldman Sachs, so he really understands the economics part and the health part, where this panel was asked to forecast what the impact of increasing antimicrobial resistance might be on our society, on the health sector, and also in economic terms. So the results of this report are quite scary. As you already indicated in the introduction, if we don't act now and we don't come up with ways, either through drug development or new diagnostics or other approaches, to combat antimicrobial resistance. One of the forecasts is that we'll have 10 million global annual deaths because of antimicrobial resistance, and that's actually more than cancer or cardiovascular diseases. And Jim O'Neill, being a health economist, he couldn't resist attaching an economic picture to this. And the estimate from the report is that this corresponds to an aggregate $100 trillion in economic cost by 2050. So those numbers are quite scary. I want to use this particular slide as a framework throughout my talk. And I want to use this to illustrate the vicious cycle that describes how we develop new antibiotics and how those antibiotics very rapidly become more and more useless because of the rapid evolution of resistance in simple bacteria. And the cycle starts here, where we as a society, pharmaceutical industry, hand in hand with academic researchers, develop new antibiotics. Those are brought on the market, being used in a clinic and in primary healthcare. And often that use is highly appropriate. These drugs are still saving lives. But as I'll show you later in my talk, often this use is inappropriate, where there's an overprescription of antibiotics and an overuse of antibiotics in the agricultural sector. This inappropriate use, this overuse of antibiotics, results very quickly in the emergence and the selection of resistant bacteria. These bacteria now aren't being able to be treated by those antibiotics and will very rapidly spread throughout the health system, in hospitals and the community. This results in higher risk of treatment failure. Think of simple surgical procedures like dental procedures, like hip replacements, that now are commonplace and very routine, but in the absence of appropriate antibiotics to treat bacterial infections that often are the consequence of such routine surgery, those surgeries are not routine anymore and become extremely high risk. And of course, the vicious cycle then results in the fact that this drives us as a society to develop new antibiotics. But as the Americans say, in a way, this is really kicking the can down the road, giving rise to resistance of those new antibiotics. I want to start my story by telling you a few things about the molecular work we do, trying to understand how bacteria deal with antibiotics, and really starting from the starting point, how bacteria treat their DNA, how they copy their DNA, and how they repair their DNA. So first, the basics. So I think most of us in the audience are aware of what DNA is, the blueprint of life. And I think it's important to realize here that every organism on Earth, animals, plants, but also microorganisms, they each have their own DNA genome, long strings of A's and C's and T's and G's that encode for the proteins and other cellular components that these organisms need to survive. Every organism, at some point just before cell division, just before one cell splits into two, every organism needs to copy their full genomic DNA into two identical copies, so that the two daughter cells carry each with them, again, a full identical copy of the genome. So this process, this copying, this duplication of the DNA is called DNA replication. And this is actually one of my earliest research fascinations, to understand how DNA replication works. So from a simple cartoon point of view, you have a double strand of DNA, the famous double helix, that is split into two, resulting in two single strands of DNA, on which complicated protein machineries then synthesize on both strands new DNA. So you essentially end up with two identical double helices, copies of the original DNA. This DNA replication is a fascinating process, and I want to walk you through a couple of numbers. And I promise you, these are the only numbers and the only maths you will see in my presentation. And I want to give you a sense of scale. I want to sort of estimate what the amount of DNA replication is in our body, just for ourselves, in our lifetime. And I have a couple of numbers listed on top of the slides, and a couple of numbers you have to sort of take for a given, we have roughly 100 trillion cells in our body. So that's 10 million times 10 million. Each cell divides on average during our lifetime 50 times. And I'm a physicist, so I'm taking a bit of a shortcut here. If you're a biologist, you would say, well, that's actually not quite true. Some cells divide more rapidly, other cells divide more slowly. For example, our brain cells stop dividing essentially when we're 20, 25 years old. And the cells in our hair follicles continue to divide throughout our lifetime. Realizing, looking at my own image in the screen, that point already stopped a while ago for me. But if you take the average of all cell types, then you have an average of 50 times per lifetime. For human cells, each cell contains 2 meters of DNA. So you essentially have to copy 2 full meters of DNA per cell division. Now let's go through the numbers. If you multiply those numbers, you'll find out that you are copying 10,000 trillion meters of DNA throughout your lifetime. And this is a number that's too abstract, it's too big. If you put this in normal people units, you'll realize that this is exactly 1 light year. So during our lifetime, we copy 1 light year of DNA per person. It's an amazing number, I think. But the most amazing part is that most of us will not get any diseases that are related to mutations in the DNA, for example, cancer. And this tells you that DNA replication is a highly accurate process with only roughly 1 mistake made per 1 billion bases or letters that need to be copied to get a full duplication of genomic DNA. So I think those numbers are amazing. So partly because of those numbers, I grew really fascinated by this process, DNA replication, a number of years ago. And as a physicist by training, having moved into biology later, one of the goals that I set out for my research group 10, 15 years ago is to develop physical tools using lasers, using microscopes, using sensitive detection techniques to visualize at a single molecule level how these DNA molecules are being replicated. And what you see on the slide here is in the top left corner is a little movie that is an artist's impression made by an Australian actually from the WeHi in Melbourne. That is actually fairly accurate depiction of how we think this process works. And what you see there is obviously a few strings of DNA molecules. On the left-hand side of this movie, you see the parental DNA, the double helix that is going to be copied. And what you see here is a massively complicated system consisting of multiple protein components that in a highly orchestrated way race through that DNA molecule, do all kinds of really complicated protein things, and as a result produce 2 identical copies of the double helix. So it really was our goal a number of years ago to visualize this process in real time at the single molecule level, to make molecular movies. And to quote one of my idols, Yogi Berra. Some of you might have heard of him. He's a former baseball player in the US, and probably one of the most famous baseball players, and later on a coach. And he has a whole catalog of beautiful quotes. And famously, he said at some point, "You can observe a lot by just watching." And that's one of my main philosophies in my research career: develop tools to visualize processes at the molecular level and just see what happens. And you'll find out an amazing amount of detail. So one key point that I already mentioned in passing is that we set out to visualize these processes at the single molecule level. And I can tell you it is quite an engineering challenge, sitting behind the development of super sensitive microscopes that allow you to see molecules at the single molecule level. And remember, traditionally, chemists and biochemists have been doing those kind of experiments using flasks and Erlenmeyers and tubes filled with gazillions and gazillions of protein molecules at the same time, doing those reactions all at the same time. So this video shows you, I think, very nicely what the advantage is of visualizing in real time how a process works at the single molecule level. So you see the tortoise and the hare here. And of course, if you do an ensemble experiment, if you look at a gazillion of those races, you will of course expect the hare to win on average. but you will have individual reactions where that might not happen, like this one. And then it's really important, or for any of those reactions, to understand what happens in real time. And in this particular case, you understand the reason why the hare isn't winning, because the hare is pausing at times, allowing the tortoise to leisurely walk to the finish line as a winner. And this embodies the philosophy of watching processes, molecular processes at the single molecule level. So I want to give you an example of one type of experiment to give you a bit of a feel of how that looks like. So what you see here on this slide is a fluorescence microscopy movie on the right-hand side, where we have a field of view, a part of a sample of a specimen that we can see that is 0.1 millimeters by 0.1 millimeters. And we take individual DNA molecules and we almost like a cloth line, string them along the surface of a cover slip under the microscope by attaching the DNA molecule on each end to the glass surface of a cover slip. And in this movie, we use a fluorescent molecule, a dye, a stain molecule, to visualize where the duplex DNA is. And you can see all these beautiful little lines. And if you look closely, you can see them jiggling. That's really because of the nanoscopic movement of objects is always very random and violent, and everything moves around. And we use those stretched DNA molecules almost as a racetrack to see how these replication protein machineries move along them and copy that particular DNA molecule. So this is a slide that shows you the complexity of the biochemistry of those protein systems that copy the DNA. So what you see on the right-hand side is a cartoon depiction of the same kind of protein complex that I just showed you in this movie a few slides ago, where you see a couple of brightly colored blobs that take the double helix that it needs to copy, and now in this case sitting on the right-hand side, and as a product, it spits out two identical copies of the double helix. And this slide is only showing you the complexity of the biochemistry that we're dealing with. On the left-hand side, you see a complicated laundry list of a large number of acronyms. In this case, I think we have more than 35 of these different acronyms. And each acronym stands for an individual protein that we in the lab need to physically put together to establish these complicated multi-protein complexes that we can then assemble on the DNA molecules that we have strung along the surface of our microscope cover slips. And then we're in a position to visualize them in real time, how they copy the DNA. And I'll show you this set of movies here, where you see on the top side, especially the top left side, one of these DNA molecules stretched along the surface. This particular movie is again using this dye molecule that stains the entire DNA molecule. But in this case, we have a replication process, a replication complex starting here. And this movie is looping. And you see this blob moving from the left to the right. And at the bottom, you see a different visualization of this process. The time runs along the y-axis here, and the position is along the x-axis here, and you see this blob moving along the DNA as an indication that you're producing DNA there as the complex moves along the DNA. And one of the cool things we can do, shown in the two columns here in the middle and on the right, is that we can fluorescently label, put a specific marker that gives off light a specific color, attach that to one of the individual proteins in the complex, and we can then see how these individual proteins move along with the complex. So on top here, you see pol epsilon and pol delta. It's not really important in terms of details what I do, but those are two key components of this DNA replication machinery. So this kind of technology has allowed us, since the last 10, 15 years, for the first time in the world, I might add, and of course I'm a bit proud of that, we're able to see these processes at the single molecule level, and we've learned a tremendous amount of molecular information of how these DNA replication complexes actually work. So what does this have to do with antibiotics? At the University of Wollongong, in the institute that I'm directing, Molecular Horizons, we have a wonderful team of synthetic chemists, medicinal chemists, and molecular biologists and molecular biophysicists like myself, who are looking at DNA replication in bacteria as a target for novel antibiotics. And remember, in the vicious cycle I showed you before, this development of new antibiotics is a key point. But I do want to make a couple of comments here on the development of new antibiotics. What you see here is a timeline starting in the late 20s all the way until now are two things. You see a couple of bright blobs indicating when different new classes of antibiotics have been discovered, with the year indicated in them. And above that, you see for the same colors of circles corresponding to the same classes of antibiotics, you see the years in which we observed resistance in the clinic for the first time. And this is something Alexander Fleming in his Nobel Prize lecture in 1945 already warned for. That, look, he said, every time you come up with a new antibiotic, you will see resistance developing in the clinic very quickly. Bacteria are very clever, they evolve very fast, and you will see resistance. So in some cases, for example, here, you see resistance in the clinic literally years within the introduction of that antibiotic into the market. And sadly, it has been a couple of decades since we've really discovered an entirely new class of antibiotics. So this is a problem in the antimicrobial resistance space, that we really have a lack of new drugs. So me and my colleagues are thinking about DNA replication as a novel target for drug development. So that's a story hopefully to be continued. I want to switch gears a bit now and then focus on what might happen if you have developed a new antibiotic and you release that antibiotic onto the market and you start to use it either appropriately or inappropriately in the health sector and the agricultural sector. And that is the problem of emergence and selection of resistance bacteria. So most of you remember from either high school or university that evolution is a process where you have a large population of organisms, be it bacteria or humans, and then by chance, some of them might have a mutation that gives them a competitive advantage, and combine that with what we call selection pressure, survival of the fittest, if you wish. That mutation then might become dominant and take over the population. So this is evolution, of course, described in very simple terms. With bacteria, it's not quite that simple. So what you see here on the top is schematically indicated here on a plate, a bunch of bacteria, the lightly colored ones and a dark colored one, where in this case, there happens to be a dark colored bacterium that is resistant to a certain antibiotic. When you apply the antibiotic, then classical evolution would tell you that it kills all the other bacteria and only this one dark colored bacterium survives and takes over the population. However, our lab and other labs in the world have started to realize that antibiotics do something else. They not only kill sensitive bacteria, but they also trigger processes in bacteria that speed up evolution, that cause the bacteria to increase their mutation rates. So the act of using antibiotics itself puts the resistance evolution process in fast forward. And schematically, what it means, you might have bacteria that are all sensitive to the antibiotics. You apply the antibiotics, and that process itself might make them resistant to antibiotics. So there's a catch-22 here. You need the antibiotics to treat infections, to kill bacteria, but by doing so, you increase the risk that they become resistant. So this is really the crux of the AMR, of the antimicrobial resistance problem. So what I'm going to show you here needs a bit of explanation. This is an experiment that a very talented research fellow in my group did a number of years ago, and that has really spun off a new direction of research in the lab. And that is what we call experimental evolution. So what we did is we made teeny tiny structures through which we can flow solutions of bacteria and mixtures of antibiotics. And we then exposed bacterial populations to concentrations of antibiotics that gradually go up and up and up, and see how those bacteria evolve. So I'll talk you through this movie on the left here. So first of all, what you see here in the upper left corner is the time in hours and minutes. So this is what we call a time-lapse experiment. You're looking at what we call a microfluidic flow cell, where we have bacteria that give fluorescence. Bacteria typically don't do this naturally, but we change the bacteria, in this case E. coli, a very common lab bacterium. We change the bacteria so that they give off a bit of fluorescence so we can more easily see them and understand how they grow. You see two channels, two reservoirs, one on the left here and one on the right. In this reservoir on the left, we have no antibiotic. On the reservoir on the right, we have an antibiotic. And these reservoirs are connected, and you hardly can see it in the movie. They're connected by teeny tiny channels through which the bacteria can crawl and grow towards the reservoir that has antibiotic. Now, the thing is that this side here has food in the solution. And as the bacteria grow, they run out of food, and that drives them to really search for new food, in this case, in the reservoir that has an antibiotic. Now, what the amazing thing is, and you see the movie looping, and you've probably noticed already, that when the bacteria grow into this reservoir that has antibiotic, they grow less quickly, they're less happy if you wish, but at some point of time, an example here highlighted in the red box, you see the bacteria growing very slowly, very unhappy, but all of a sudden, you see an explosive growth. And this is really the hallmark of these bacteria at that moment in time, mutating their DNA very rapidly, and apparently one of these bacteria in that red box hitting a mutation that is making it less sensitive to the antibiotic. And I really want to point out the time scale of this experiment. It's less than 2 days. And you can see when you look carefully at the movie that the process of gaining these mutations that make the bacteria less sensitive to antibiotics takes only several hours. So under lab conditions, this evolution process can go scarily quickly. So just as a tiny experimental follow-up, what we started to do then is develop the technology to visualize individual proteins inside living bacterial cells. And you see a couple of movies here where we've developed technology on different time scales. On the left-hand side, hours. On the right-hand side, super fast imaging on seconds, where we can see actually the movement of these individual molecules inside bacteria. So what we're now doing, and I can't really show you the outcomes yet, but what we're now doing is combine all those experimental approaches to experimentally evolve bacteria and make them become insensitive to antibiotics in those microfluidic flow cells, while observing what the individual proteins of relevance do inside the bacteria, in particular the proteins that put in the mutations, and see how they behave. And this would allow us, for the first time ever, I think, to couple the molecular details to what we biologists call the phenotypic details, meaning that you see from the outside that bacterial populations become insensitive to antibiotics. And we have strong hopes that this will allow us to much better understand what this evolution process really is and how it works. And of course, as a consequence, how we can better deal with it. I want to take a few minutes here to do a bit of promotion and pitching. As Hugh already indicated, we started a few years ago at the University of Wollongong a fantastically new initiatives where the university spent a lot of money in investing in research infrastructure that has, again, as a philosophy of visualization and molecular characterization, where we established a new institute called Molecular Horizons, where we want to use visualization approaches really as a technology driver for discoveries in the life sciences. You can see a picture of the pretty new building we have. It opened in January this year, sadly only a month before COVID hit. So we haven't had an opportunity yet to throw a big party and have an official opening, but that's something we hope to do very early next year. And one of the exciting things here is that we do a couple of things in the institute. We bring together chemists, biologists, and physicists. I hope this far in my talk, you've got an appreciation for why that particular combination of disciplines can be really powerful. And as I said, we want to use advanced microscopy to visualize the processes of life. And there's a couple of disease focuses in the Institute, including AMR, motor neuron disease, cancer, and Alzheimer's, and many more. One of the flagship developments that we have in the Institute is a new facility where we have started to use cryo-electron microscopy. And I showed you a couple of movies that are based on optical microscopy using classical lenses and classical microscopes in a way very similar to the ones you might remember from high school. But cryo-electron microscopy is a completely different beast of visualization technology. Instead of using photons, light, to visualize processes at the molecular level, we use here electrons. And the cool thing about electrons is their wavelength is so much shorter than that of light, and that allows you to see even smaller and smaller details than you can do with the most powerful optical microscope. And we started to use these really powerful cryo-electron microscopes to visualize all the structural details, all the nooks and crannies in individual protein molecules. And what you see here on the top is a typical microscopy image. It's very noisy, but if you squint, you can see these particles sitting there. They're all pretty much identical. And these are individual protein complexes. We can then take hundreds of thousands of these grainy, noisy snapshots of individual particles and use computational processes, partly driven by machine learning, to do averaging of all these individual pictures. And that gives you fantastically high spatial resolution. In this case, you see a protein complex where we have resolution of much less than 1 nanometer, which is enough resolution to see where every individual atom sits within every individual protein. And below here, you see a gallery, just to show off the pretty pictures we have of some other structures of proteins we've solved in our new facility. And the reason why this is important in the drug development process is that when you identify a protein target and you want to develop a drug that acts on that protein target, for example, the DNA replication protein, you really need to understand the structural details of how the protein looks like, so you can then start to work with the chemist to really physically be able to dock a drug molecule in the exact right spot on that protein molecule. So again, seeing is believing. We need these types of microscopes to visualize not only how these proteins work, but also how they look like in great detail to facilitate this drug development process and to be able to work on new therapies. So the last bit of my talk, I want to really zoom out and say goodbye to the molecules in the cells for a few minutes and talk about the community level aspects of antimicrobial resistance. Going back to the vicious cycle, I already told you that one of the driving forces, or probably the driving force of antimicrobial resistance, is that we overuse antibiotics as a society. There's a lot of appropriate use. Again, antibiotics are still life-saving drugs, but also there's a lot of inappropriate use. And that then in the end results in spreading of resistant bacteria in hospitals and the community. One of the key problems is that we really don't understand how AMR, how resistant bacteria travel through the community, through the hospital system, largely because we don't have access to high enough quality and granularly enough data. This is really a data problem where we have started thinking with a group of highly talented people and collaborators, how can we think about improving the data ecosystem? How can we integrate data streams and fill data gaps to better know how AMR travels through the community and to have predictive value, so we can almost like a weather forecast start to predict where and how and when even AMR might go up in a particular region, so resistance for a particular drug and what have you. So the bottom line is this: drug development takes time. Development of Ultra-fast diagnostics takes time. Drug development in particular, when you have a new antibiotic, it takes 10 to 15 years because before you can push the antibiotic through clinical trials and put it on the market. With the speed of rise of resistance in society, there's a critical need to better manage the resources, i.e. the antibiotics we still have now. So we have to keep putting resources and money in developing new drugs and therapeutics, but we have to also able to work on technology that allows us to better manage the resources we still have. But the key problems here is that there is presently no large-scale community-based cohort study that characterizes AMR and provides a test bed for holistic interventions to slow down the use of antibiotics. And as I said, we have really no solidly integrated health data ecosystem that allows us to understand how resistance travels to the community and how to respond to it. So I moved to Wollongong 6 years ago. Wollongong is part of a region called the Illawarra Shoalhaven. And not only is it a beautiful place to live, it's also a wonderful place for population health studies. The Illawarra Shoalhaven is really a microcosm of Australia. When you look at, for example, rural versus urban populations, when you look at census data, when you look at Aboriginal communities, it's a beautiful, diverse area that is a really perfect mimic of the whole of Australia. So what we have set out to do is to look at the Illawarra Shoalhaven as an AMR testbed. Also, what we like to call it is the Framingham of AMR. Framingham was really the first large community-based cohort study in the US, in Framingham, Massachusetts, in the 40s of last century, which became essentially the testbed where all our knowledge, or a lot of our knowledge on cardiovascular diseases and lifestyle factors came from. So that has been an amazing success. And we have a fairly ambitious plan to make the Illawarra Shoalhaven into the same for AMR. So we started teaming up with a lot of fantastic people, in particular a team from UTS CSIRO and the University of South Australia, to form a consortium that we call Outbreak, which has been funded by MRFF Frontiers Stage 1 funding. And we're now waiting any day now to hear whether we've been successful for Stage 2 funding, where we can roll out our plans really at scale and really make a difference in how we as a society would deal with AMR. So essentially what the Outbreak Consortium does is track, trace, and tackle. As you see here on the bottom of the slide, we want to produce and integrate data to track how AMR travels through the community. We want to use those data to understand and predict AMR, trace, and we want to use this data, and this is the key point really, to allow people who prescribe and use antibiotics, like doctors, like veterinarians, to prescribe and use antibiotics with more precision, knowing those data and understanding, for example, region-specific resistance rates. So this is the stewardship part, the tackle part, where we really can use data to minimize the use of our antibiotics and only use it when needed. So what we do, we take a One Health approach. We not only look at the health sector, but we also look at the agricultural sector. We look at the environmental sector, and we really very holistically try to bring this together, feed data and knowledge, data from a number of existing and new health systems. We feed this into essentially an AI, machine learning driven knowledge engine that will give us predictive value of how and where and when AMR travels through the community. And this is a couple of examples how end users of antibiotics, for example, GPs, for example, hospital administrators, public health officials, might use these data. And I want to give you one example for the GPs. When a patient sees a GP and wants to have an antibiotic, typically GPs prescribe according to standardized prescription guidelines called the therapeutic guidelines that essentially take a blanket approach. Those guidelines are the same for every GP in Australia that give the GP, based on clinical symptoms, a couple of suggestions for antibiotics to use. Well, we've just started to realize, again, in the Illawarra Shoalhaven, that there are huge variations in local resistance rates. So what you see here is data that we just got actually a few weeks ago, where you see the Illawarra Shoalhaven split up in different postcodes. And we started to look at the resistance rates for different antibiotics, in this case norfloxacin, which is a fairly popular antibiotic. And we noticed that in the different postcodes, the resistance rates are varying between 8%, meaning that 8% of the infections is resistant to this antibiotic, all the way down to 2%. And the scary part, when you zoom in the map here, and these are two postcodes just north of Wollongong, that two postcodes, literally 2 kilometers apart, have a 3-fold difference in resistance to norfloxacin. And this is based on urine samples and for E. coli. So this is the type of sample you would see with patients with UTI symptoms. And the statistical significance is extremely strong of our data. So this kind of information might allow a GP working in postcode 2518, where you have high resistance, to not pick that particular drug, but instead use another drug that has lower local resistance rates. So really the essence of Outbreak is to integrate all those data sets and arrive at a much more person and location specific prescribing of patients, so that you really have much more granular risk profiles around patients, or in the agriculture around the use of what antibiotics for treatment of cattle, for example. So our mission is really to build technology, platform technology that is driven by AI, fed by data and knowledge, to act now, knowing that we don't have time to wait for new drug development to solve the AMR problem, and manage the resources we have. And I want to conclude with just one statement by the World Bank, that action on AMR constitutes one of the highest yield development investments available to countries today. We did an economic study that we published 2 weeks ago with the Outbreak Consortium, where we realized that already the cost related to bacterial infections causing UTIs is already close to $1 billion per year for Australia. And if we don't act now, stemming these resistance rates, that might very quickly become almost $2 billion within 10 years from now. So there's a real also financial imperative here to act, plus of course, making sure our communities stay healthy. So I've gone full circle, I think. I told you some molecular things. I showed you some cellular movies that gave us more insight on how resistance evolution works. And I showed you how I think we can use those philosophies at the community level and integrate molecular level with community-based approaches, and really arrive at holistic approaches to solve these really, really tricky and big problems. And with that, I want to conclude, but not before thanking, of course, everybody who I work with and all the people who I've worked with in the past, the students and researchers in the lab, current and past, the wonderful collaborators in Australia, overseas, supportive colleagues here at the University of Wollongong and elsewhere, and of course, the wonderful funding agencies who are really critically important for allowing us to do this kind of work. And of course, I want to thank you for having me here, and you, of course, and happy to answer any questions you might have.
Thank you, Antoine. It was a fantastic talk, and it actually really inspiring to see what you've put together at Wollongong. You know, and yep, I'm definitely going to come down and visit, and when your building gets opened and things like that. Look, I've got to ask some naive questions, I guess, here. I mean, what does the pipeline for new antibiotics look like? And also the thing that troubles me a little bit about this area is, and maybe again, it's just me being naive, is it doesn't seem to be systematized in the way that other kind of vaccine type drugs or this sort of thing seems to be in development. And is there a prospect of, with the work that you're doing, of systematizing the way that antibiotics is developed into the future?
Yeah, it's a wonderful question, you. I'm sad to say that I think the biggest barrier in antibiotics development is a monetary one. If you look at current antibiotics in the pipeline, and there's a couple of really exciting ones. There's a couple of them that have passed phase 3 clinical trials there's a couple of companies that have been started, mainly US-based, that started putting these antibiotics on the market. And if you Google for a few minutes and look at their stock prices, they all crashed. These companies are simply not viable. And of course, naively, you would think, but why, right? There's clearly a need for new antibiotics. So the problem is that there is no real financial incentive for pharmaceutical companies to fully develop an antibiotic through the very expensive clinical trial pipeline, simply because we still have very cheap antibiotics. Antibiotics are the cheapest drugs you can imagine. And then, of course, one sad realization is that you treat a patient with your drug and then they're done. You don't see them again. So there's a couple of lacks of incentives from the pharmaceutical industry point of view that don't make it a viable proposition. And the biggest pharmaceuticals, I think almost all of them, in the last few years have explicitly stepped out of the antibiotics market. So I think this requires, from a governmental point of view, and this is also one of the recommendations from the O'Neill report I showed you in the beginning, to incentivize antibiotics development by government incentives. And this is, from an economics point of view, a really tricky one.
So let me just follow up with that. Because Scott, can you just check for me whether I'm actually looking? I've sent you a number of questions, whether anyone is actually asking questions or whether I'm on the wrong chat channel. Please, Scott, thank you. So Antoine, just let me follow up on that a little bit. So is any government actually doing this at the moment? Is any government proposing to genuinely fix this kind of market failure?
Yes. In Europe, there's a large initiative. There's a large initiative happening since a few years where industry and government have started work together to facilitate the screening and characterization of large numbers of candidate drugs, because that, of course, is a costly process. And of course, we're all familiar with the valley of death, that long period between something being developed on a university campus on the one hand and it actually being taken up by a company as a commercial viable thing on the other hand. So there's a couple of things happening in Europe in particular, but I don't think there's really a large higher level incentivization structure yet.
Okay. And then the systematization of developing antibiotic. Is that a prospect?
Yeah, absolutely. I think a key thing to realize is think about the molecular details. And a reason why most antibiotics have caused resistance very quickly is that they hit one particular site on a protein. And often you only need one or two mutations in the protein to make that drug not work anymore. So one approach my colleagues here at Wollongong have taken looking at replication is that you need to identify a point on the protein that itself interacts with many other critical proteins. So you only then can mutate when you have a simultaneous mutation in all the partner proteins that physically connect on the same point. And it's a bit of an indirect way of answering your questions. What I think we need to do is to come up with general rules around what sites to target that would make resistance evolution less and less likely, and then develop the technology to screen those, but really focusing on those multi-protein interaction sites. Okay.
So actually, I have managed to get some of the questions through now. So the first one is from Gillian Skinner, who from memory used to be the Minister for Health. The question is the use of data analysis the main difference between Outbreak and other AMR projects? And how much more important does this make Outbreak?
I think there's a couple of key differences. Outbreak takes an approach not just integrating data and using that data, again, supported by machine learning to make predictive scenarios. It also relies on producing data by integrating the population public health part with the One Health component. So really looking at the agricultural and and the environmental sectors as a key component. Because something I really didn't talk about is that we do know that a lot of these resistance genes inside these bacteria, the particular bits of DNA that cause a bacterium to be resistant, are jumping back and forth in a way between the environment, animals, and people. So what you really need to do is, as I said, come up with better data integration and use and develop the technology to get these predictive values, but also roll in the One Health component. And I think one thing that makes Outbreak unique is that we focus on platform technology. And then, and this is a bit of a commercial strategy, develop that platform technology so that other commercial entities can start to develop products that are very comparable to the Apple App Store. So you have the platform technology sitting there,
Understood.
You have the data sitting there, and other people can then develop end products.
Yep, that's a very good approach, I think. There's another question here from Tim Boyle, which is timely. What are your thoughts on the role of AI protein folding determination, like the DeepMind platform that was, or the DeepMind result that was announced this week? Do you think this will help in solving some of these problems?
Yeah, absolutely. I think most of us have seen on social media and in the news this announcement that there's a significant step being made in protein folding. And this is a problem people have been working on for, well, I would say almost a century, but in the last few tens of years in particular, supported by high performance computing, because protein folding is one of the most challenging computational problems you can find. And so there AI is going to play an enormous role to really have an objective and unbiased way of identifying themes and shared processes across different folding problems. So to give an answer directly to your question, in terms of drug development, I wouldn't be surprised that within the next few tens of years, we will be in a situation where you can actually fold proteins and, more importantly, predict how it might interact with large numbers of hypothetical small molecule structures.
Yeah, that's really interesting. Okay, Antoine, thank you so much for a really engaging talk, and there's some fantastic work that you're doing down there. I think the Magic Chief Scientist mail system got you a book and a bottle of wine. Excellent, wonderful. This shows we work in 3D as well as 2D. And that brings to a close the series for this year. It's been a fantastic year. I think we've genuinely heard some really amazing talks, very relevant to the long series of issues that we've had this year, to be followed by the first presentation next year, to be given by the 2020 New South Wales Scientist of the Year, Eddie Holmes, a very well-known virologist from the University of Sydney. And we hope that his posting of, in fact, the DNA model for the coronavirus will actually yield enough of an outcome before the first talk next year that we might actually be able to meet in person for the first time. So that would be great. So we'll be in touch shortly with everyone here about where the seminars will be next year. And I want to again thank Antoine for a wonderful talk, and I'll be down to visit in the new year. Thank you, everyone. Bye.